Good afternoon and welcome to today's webinar sponsored by Rice University's Baker Institute for Public Policy, Center for Health and Biosciences, and the Immunization Partnership. Thank you for joining us online. My name is Kirsten Matthews. I'm the Baker Institute Fellow for Science and Technology Policy. I am hosting today's session on COVID-19 pandemic titled COVID-19 Update on Vaccine Development. The discussion today will have a few questions that we've prepared in advance from the speakers based on recent developments and questions submitted online during the event. Our featured guests today are Rekha Lakshmanan and Dr. Peter Hotez. Rekha is a contributing expert in the Center for Health and Biosciences at the Baker Institute. She is also the Director of Public Policy at the Immunization Partnership in Houston, Texas. Her work involves development and implementing public policy strategies to improve Texas vaccination rates, advising states and organizations on building grassroots networks, and teaching constituents how to communicate with lawmakers. Rekha holds a master's in healthcare administration from Texas Women's University and a BA in government at the University of Texas at Austin. Dr. Peter Hotez is a Baker Institute Fellow for Disease and Poverty. He's a dean for the National School of Tropical Medicine at Baylor College of Medicine. He's also a professor of pediatrics and a professor of molecular biology and microbiology. He holds an MD and PhD and is internationally recognized as a physician scientist with expertise in neglected infectious diseases and vaccine development. I first want to thank both Rekha and Peter for joining us today.
Thank you for having me.
Our first set of questions, we're going to talk a little bit about vaccine development to start with. I think it's a good way to kind of bridge into talking about specific vaccine developments for COVID. And I'm going to have my first question to Rekha. What is really the process of getting a vaccine approved by the FDA? Can you kind of walk us through developing a vaccine, kind of starting with viable candidates, going all the way through getting approved by the FDA and when we actually see it in the clinics?
Sure. So, I think it's important to remember that, vaccines go through a very time-intensive and rigorous scientific process before a vaccine even becomes available to the public. And from a time context, it can take anywhere from 10 to 15 years, sometimes even more, to study and research a vaccine candidate and then make it available to the general population. And even though we have vaccines that go through this trialing process, there's no guarantee or assurance that we're actually going to get a viable vaccine candidate out of all of these trials. An example would be an AIDS vaccine. Researchers were working on an AIDS vaccine for many, many, many years, and unfortunately, at the conclusion of the trials, the vaccine wasn't viable. But from a phasing standpoint, there's a preclinical phase where preclinical studies are done, and they can use either tissue culture or cell culture or animal testing to assess the safety of a vaccine candidate, looking at the immunogenicity, seeing like what kind of immune response would this candidate provide, and how well it provokes an immune response. And so if animal subjects, for example, are used, it can be either in mice or monkeys. And this is a really important phase because oftentimes many vaccine candidates don't even make it through the preclinical phase because it didn't elicit the right immune response. However, if a candidate does successfully pass the preclinical phase, then there are 3 additional phases a vaccine candidate goes through. Phase 1 simply includes a small group of people. It can be anywhere from 20 to 100 individuals. And the questions scientists are trying to answer in phase 1 trials is, is this vaccine safe? What kind of immune response is generated, from a person who's given the vaccine? How much vaccine or what is the dose needed to elicit an immune response? And oftentimes phase 1 trials last anywhere from several months, but typically less than a year. And as we know, in terms of what is in the current pipeline for COVID-19 vaccines, a lot of vaccine candidates for COVID-19 are in this phase 1 trial. Now, if scientists, have their questions answered, in terms of, is this vaccine safe and does it elicit an immune response, then it moves on to phase 2, which includes a larger pool of volunteers. And again, the same question, researchers and scientists are asking is this vaccine safe? What are the side effects that may emerge when someone gets this vaccine candidate, and how does the individual respond to the vaccine? Once those questions are answered satisfactorily, then it goes to phase 3, where the pool of volunteers again gets bigger. So now it involves hundreds, if not thousands, of volunteers. And what phase 3 does is to really, again, assess the effectiveness of the vaccine, but also to assess vaccine safety in a larger group of people. Sometimes there are certain rare side effects that might not surface in the smaller groups of subjects tested. And so phase 3 allows for some of those potential side effects to emerge. And then once phase 3 is successfully performed, that's when the vaccine can go to the FDA, and then the FDA will evaluate the data again for safety and effectiveness. And once those questions are answered, then the FDA will make a decision as to whether to approve a vaccine for public use or to not approve it. I think it's also important to just remember that, once a vaccine is licensed by the FDA, that vaccine is still continuously monitored, for safety and for efficacy through a variety of safety monitoring systems, not only here in the US, but globally.
So now I'm going to kind of bring Peter in at this point. So we kind of have an idea of the process, but we've been hearing a lot of different figures about the current vaccine progression for COVID-19. Some are even indicated they think they could have it available as soon as September. Others are saying a year, 18 months. That's something we generally hear from Dr. Fauci, from the NIH. How optimistic are you about the potential of getting a vaccine this fall? What do you think about these predictions of the early ones, or which one do you think is more realistic?
Yeah, so I think Rekha gave a great overview. There's a lot of confusion about what's really going to happen and what we're hearing from the pharma companies and some of the even statements coming from the White House Coronavirus Task Force. Here's what's happening. There's going to be at least 7 or 8 and probably as many as 15 or 20 candidates that go into clinical trials. And hopefully our vaccine that we've developed at the School of Tropical Medicine at Baylor and College of Medicine and Texas Children's will be one of them. So the first ones went into phase 1 trials in March. That was the Moderna vaccine. There's going to be lots of others that will follow, and everything will proceed in a staggered fashion. And most likely the first candidate, which will likely be the Moderna one, since that was the first one that started in phase 1, will go into phase 3 trials towards the end of the summer. And the estimates are it'll take at least a year and maybe longer to collect enough data showing that the vaccine actually works and the vaccine is safe in a large phase 3 clinical trial with around 25,000 patients to know whether we can start releasing some of those vaccines to the public after licensure. So the earliest that I estimate we'll see any vaccine would be by the 3rd quarter of 2021. And even that would be a world record. The world record being, some people call off it says mumps, which is 4 years. Now you're hearing other things. You're hearing certain vaccines will be available by this fall or by the end of the year. I don't see a path by which that happens. What will happen is we will have some of these vaccines manufactured potentially by then. Because part of the way we're accelerating this is once we do have some vaccines that we know are safe and effective in a phase 3 trial, a large phase 3 trial, then we can begin introducing the vaccine into the United States. And the only way to do that is to have it manufactured from the beginning. So the phrase that you're often hearing from Dr. Fauci and others is manufacturing at risk. At risk means we might make several of these vaccines scaled up manufacture, even if we don't know if they work or they're safe. So the point is, if they're shown not to work or not to be safe or have some issue, we're going to throw them away. So that's the at-risk part. So if you're not constrained by resources, in theory, you can do this. This is the first time we've ever done it. So I think that's one of the ways you're seeing acceleration, not by compromising safety trials or compromising efficacy studies, but by doing things all around it. Another thing that we're doing to accelerate this is to look at multiple candidates at the same time. So all of the vaccines, including ours, work by inducing an immune response to the spike protein of the virus. And you've seen those cartoons with the spike sticking out of the virus, and that's what binds to the host receptor. All antibodies work by inducing antibodies to neutralize the virus, that is, prevent the virus from attaching to the host tissues. And they're all using different technologies. Some are using recombinant protein technologies like ours, others inactivated virus or live attenuated viruses like the Merck vaccines or DNA vaccines or mRNA vaccines or adenovirus vectored vaccines. And the reason you're doing that is to give you more shots on goal with different technologies. So you're more likely than not to have something out the other end, because 90% of vaccines fail to make it all the way through clinical development. So if you want 2 or 3 vaccines at the end of this, which is what the hope is, you do the math, you need 20 different candidates to go through. So that's another way we're accelerating it. Then we are doing a few things like consolidating some of the trials. So typically in a phase 1 trial, you would do that in healthy adult volunteers in the ages of 18 and 45. And then for your phase 2 study, you might do expanded studies on alternate populations like older individuals. And what we're doing here is trying to consolidate some of the phase 1, phase 2 trials to start looking at older individuals from the very beginning in phase 1. Also looking at doing the formal toxicology testing in parallel with the phase 1, if it's a technology that has been shown over and over again not to create a safety signal. So there are a lot of things you can do to accelerate timelines without compromising safety or getting information on the efficacy. So I think that looks like a sound plan, but it also means we're not going to have a vaccine by the end of the year or in weeks or months. And unfortunately, a lot of the imagery and language around these processes have created a lot of confusion. So you're seeing a bunch of press releases that verge on being irresponsible by some of the biotechs saying we'll have a vaccine in weeks or in days. What they're really meaning is they can, get them manufactured or get them produced at small scale quickly. And then you have the White House using that imagery of Operation Warp Speed, which creates a lot of problems. And as we'll talk about with the anti-vaccine movement, but I think there's quite a bit we can do. And if all of this works and the stars align and we do have a vaccine at the other end in, say, a year and a half from now, that may change the way we think about doing vaccine development going forward, because we will have other pandemics and other epidemic threats.
So this is a great transition, because you already started hinting about some of the ones that everyone's been talking about in the media. It's been reported, I think, from the World Health Organization that we have like over 110 candidates for vaccines. And you mentioned a couple dozens that are going to be through the trials pretty soon or in trials. But 3 of them have actually kind of been in the media, at least in the US, quite significantly. The one from Oxford University, the one you mentioned from Madero, and one from Invivio. They've received a lot of press, and I was wondering, because they also mentioned that they're kind of different technologies than we've used before. So if we start with one at a time, the one that you mentioned already, the Madero, it's described as a mRNA-based vaccine, an RNA-based vaccine. Could you kind of walk us through what that means as an RNA vaccine, how that's different from what we use in current models for vaccines, like for polio or for the flu? And then walk us through a little bit of the data that they've released so far and your thoughts on it.
Yeah, so if you look at some of the first vaccines that we used in the United States, like the polio vaccine, that was done by growing up the polio virus and then inactivating it with formaldehyde, formalin, or using other systems of inactivation, and then basically tricking the immune system into thinking it was live polio. Or in the case of the Sabin vaccine, using a weakened version of the polio because they passage it multiple times through cells or animals or in some cases eggs, like they've done with with some of the yellow fever virus and things like that. And so some of those vaccine prototypes will be used for COVID-19. The next generation of that was to genetically engineer pieces of the virus as a recombinant protein, so that rather than use the whole spike protein, purify the spike protein of the virus, it's not easily feasible to grow enough of the virus up to isolate the spike protein to immunize large numbers of people. So what you do is you genetically engineer it. You put the gene for the spike protein or a part of the spike protein into an expression vector such as yeast, and then you grow it up in a fermenter, just like you make beer, but instead of releasing the alcohol, the yeast are releasing the recombinant protein. And that's what we're doing for ours. It's the same technology used for the hepatitis B vaccine that's been around for 20 years. And we like that one because it's there's a lot of capacity for making recombinant hepatitis B vaccine in multiple countries like Brazil and India. So as a global health vaccine, it has a lot of attractive features. The problem with developing our recombinant protein vaccines is it took us a couple of years to make those for SARS and MERS and now COVID-19, because you have to work around with different fermentation conditions and purification conditions to optimize it. So it does take time. In our case, we're able to look at the vaccine we made for SARS-1, and we think it'll cross-protect against SARS-2, and we've accumulated some data showing that that appears to be the case. But there's still interest in seeing if you could do something faster, and companies like Moderna and Inovio has the DNA platform, and Moderna has the RNA platform, is that once you know the genetic code of the virus, which we knew in January, you can make a piece of RNA or DNA pretty quickly, within hours or days. And then provided you figured out how to package it right to induce an immune response, you can make a vaccine pretty quickly. And that's how Moderna and Inovio have attracted a lot of investment from shareholders around that concept. So that's the idea behind it. Now, each of these technologies has strengths and weaknesses. So the strength of something like our vaccine, recombinant protein vaccine, is it's well established. Regulatory authorities are used to seeing recombinant protein vaccines. There's a lot of capacity for scaling it up in low and middle income country settings. They're scaling it up everywhere, and therefore having a widely accessible vaccine. The disadvantage of it is it takes time to produce if you haven't made it before. In our case, we hit the ground running because we made it to the original SARS-1. Now we're making one that's a second prototype vaccine for SARS-2, but that's going to take several months in order to make that happen. The advantage of the things like an RNA or DNA vaccine is you can make it quickly and you can get it into clinical trials quickly. The disadvantage is that technology's never materialized into a licensed vaccine before, in part because although it may work as well in laboratory animals, sometimes it doesn't seem to work as well in people and never has gone through licensure. Also, there's not a lot of experience in scaling up production of those. And the other piece of this is the fact that these RNA and DNA vaccines are not always the best for inducing a certain type of antibody that we know we need for coronaviruses, which are called neutralizing antibodies. So, we discovered this in the decade we were working on coronavirus vaccines, that the protection more or less correlated with the ability of the vaccine in laboratory animals to induce an antibody response, but a very specific type of antibody response, an antibody that not only bound to the spike protein, but blocked it from invading cells. That's why they use the term, so it neutralizes the virus. And we can, our recombinant protein vaccines, some of the inactivated vaccines give really high titers of that neutralizing antibody. The other ones haven't. Now, you might say, okay, that was SARS-1. Well, what about SARS-2, the one that causes COVID-19? Well, Dan Baruch's lab at Harvard Medical School has now done some studies in non-human primates working at Beth Israel Deaconess Hospital to show that just like SARS-1, SARS-2 protection also seems to correlate pretty well with high titers of neutralizing antibody, which means that developing a COVID-19 vaccine may not be all that complicated. It's a straightforward problem, and it's old school virology problem. You need to induce lots of neutralizing antibodies, and we know how to do this. We've done this with multiple other vaccines in the past, but it does mean that certain platforms will be better than others. So I think, this means that some of the old school vaccines, inactivated virus vaccines, attenuated virus vaccines, recombinant protein vaccines, those are the ones I would prioritize if you needed to induce high levels of neutralizing antibody. Some of the others, the RNA and DNA vaccines, have never really been great at doing that. They do other things in the immune response, maybe better for other types of pathogens, but I'm not sure these are going to be the best for eliciting high levels of neutralizing antibody. Now, the companies are saying they're working around this problem. They've fixed a lot of things that historically RNA and DNA vaccines couldn't do that, but we'll see. We'll see how it turns out as we get data in phase 1 trials. And same with the adenovirus vectored vaccines, like the one out of AstraZeneca, which licensed it from Oxford, or the Chinese adenovirus vaccine, but they also have not given high levels of neutralizing antibody. But we'll see. Maybe there's other ways to package it. But we're going to learn a lot as we move through these phase 1, phase 2 trials. And we'll get a much better, first, one of the things we'll learn is which vaccines are good at inducing high levels of neutralizing antibody in people, because you never know until you put it into people. And then as we go through phase 3, we may even confirm that it does correlate with protection. Because if you can develop that as what we call a surrogate correlate of protection, that's really useful information, because it may mean that all we need to do is show that the vaccine induces high levels of neutralizing antibody, and not necessarily for efficacy purposes going through all of the full phase 3. You might only need to do enough just to show safety at this point. So we're going to learn a lot. I'm pretty confident we will have a couple of COVID-19 vaccines, but it's not generally not quick business. I don't see a path by which we'll have vaccines by the fall or by the end of the year or even the beginning of next year. I don't see how we collect all of that information we need until the middle of 2021, and even that would be a world land speed record.
As kind of a follow-up question to that, though, is do you think that there's going to be any side effects of actually trying all these new vaccines we've never tried before? Have there been anything shown in animals? You said they seem fine in animals, but obviously a mouse is not a human.
I think the RNA and DNA vaccines generally have had a pretty good safety profile. I know at the highest dose, it's in a press release, so who the heck knows what really happened. Data either in a published paper or at least a bioarchive preprint server, it's really hard to know. The highest dose caused, the 250 microgram dose for the Moderna vaccine seemed to cause fever and a lot of side effects, and they're not going to look at that dose any further. I'm not so worried about that. The big concern that people have, and we're working with a group now through the NIH active program, ACTIV, is whether any of these vaccines cause a syndrome known as VAERD, which stands for Vaccine-Associated Enhanced Respiratory Disease. And something like that was seen when the first vaccines for kids were developed for respiratory syncytial virus, which was used in association with a formalin inactivated vaccine in the 1960s. It was shown that kids that got the vaccine actually wound up doing worse than the kids who didn't get the vaccine. And a year later, there were 2 deaths among the vaccinated group. And so that made people very skittish about whether VAERD is going to be a problem for other respiratory vaccines. With SARS-1, depending on the vaccine you use, especially some of the virus vectored vaccines, you did see not only VAERD, but cellular immunopathology in the livers of some laboratory animals. So there is that concern. That's how we designed our vaccine, to minimize VAERD. And we did that by just using the smallest piece of the spike protein. This is an innovation that came out of the New York Blood Center, and our collaborators Shibu Jiang and Lan Yingdu were able to show that they could cut off pieces of the spike protein, just use the receptor binding domain, still get high titers of neutralizing antibody without the VAIRD, without the vaccine-associated enhanced respiratory disease. Certain adjuvants may be worse than others. And there's pretty conflicting opinions about which type of immune response you want, which is the type to avoid. And we have had some sharp disagreements with some of the guidance documents, for instance, put out by CEPI. You've heard of CEPI, the Coalition for Epidemic Preparedness Innovation. They put out a guidance document, an expert opinion, quote unquote, document. But, we were looking at this and said, how could they put out a guidance document like this? It's the exact opposite of what the published data says. So we go back and forth and things like that. But again, one of the things we're talking about now is if there is going to be this VAIRD phenomenon, how will you pick it up in a phase 3 trial? How common will it be? And yet again, this is another reason why we don't rush vaccines, that we want to give adequate time for testing. And again, this business of calling it Operation Warp Speed is not very helpful, because it makes it seem like we're rushing things, we're giving inadequate time to attention. But again, the warp speed part is all of the stuff around the actual clinical trials, not so much the clinical trials themselves.
Okay, so I want to also kind of expand this a little bit, and I'm going to ask Rekha a question next, which is related to a concept called herd immunity. And I know that's something that other people are concerned about and looking for as an avenue to say that, yes, it's okay to go outside. Yes, it's okay to have a complete 100% normal or traditional status quo. But first, could you kind of walk us through what herd immunity is and why it's actually important and how it relates to this virus and epidemic right now?
Sure. So herd immunity, or what's also frequently called as community immunity, is when there is enough people in a population immune to a disease, thereby preventing a germ from traveling or infecting a larger group of people. The concept of herd immunity is important because, number one, not all pathogens spread with the same efficiency. So there's been a lot of talk of like, okay, well, what are the herd immunity thresholds for COVID-19? And so, and a marker, for example, is, we frequently use measles as an example, and Dr. Hotez has repeated this, many times, where measles is highly, highly infectious. And in order to achieve herd immunity, we need, about 90 to 95% of the population protected against measles through vaccination. With COVID, we're learning as we go. And according to Johns Hopkins University in medicine, they're estimating that the threshold to achieve herd immunity against COVID is about 70%. If you put that into numbers, what that means is, in a world without vaccines, as we're currently living in, that means that, over 200 million Americans, will have to have been infected with COVID in order to achieve that 70% threshold. So, by those estimates, you're looking at, maybe 2021, and then with the death rates that we're seeing right now, that means that it could result in about 500,000 deaths. Again, this is an absence of a vaccine. So, the short answer to the question is, can we achieve herd immunity against COVID-19 without a vaccine? And the answer is no, not really, unless our country is, willing to pay a very dear and devastating price. And I don't think, anybody wants to pay that price.
So I'm kind of thinking this in terms, probably because my kids just finished school. I have 2 school-aged kids, and then everyone trying to figure out what we're doing next fall. There's a lot of parents that are willing to send their kids back in Texas now because we're opening up, but others I've noticed in comments and discussions want to wait for a vaccine to show up before they're willing to actually have that kind of day-to-day, face-to-face interaction with a large population. Should we be waiting to open schools or places like that until we get an effective vaccine? Is that something that, are we going to look for a certain model where it shows that the number of cases goes down and dramatically? Is it realistic to actually be engaged like we were a year ago during the school year?
Yeah, I mean, it's a very difficult question to answer because, as we've been going through this pandemic, we've seen a lot of unintended consequences associated with COVID. And none of us could have imagined, the enormous scale and the toll the pandemic has created. So, I mean, you've got parents who have, had to learn how to be teachers at home, and kids who miss playing with their friends, either at the playground or in the parks. And I don't mean playing via Zoom. So there are, a lot of questions that are still, unanswered. The bottom line is, we don't know when a safe and effective vaccine is going to be available. So it really is a matter of finding the balance between being cautious, but also being pragmatic. But I think, your question raises, some very important points, one of which is, the need for leaders to be very transparent and highly communicative and coming up with a plan to guide the public, especially parents of school-age children, as to, how and what the schools need to do and are going to do to ensure the safety of the children and the staff that tend to those children, in the school setting. So, being clear and setting expectations up front is not going to necessarily solve the problem right now, but it will help. But as Dr. Hotez said, there's a lot of rhetoric, taking place that, we're going to have a vaccine tomorrow or, in a week or in a month. And just, as what we know about the vaccine development process, and the commitment researchers and scientists have to make sure that we've got an effective and safe vaccine, waiting for that vaccine to come out, and reopening schools, for example, is it may not be really realistic. But if you've got a good plan in place, and now that school is out, hopefully that affords some time for leaders and educators to figure out what reopening schools will look like in the coming fall.
So there is the discussion, and I think Peter's already said that he's a little pessimistic about those vaccines that are circulating in clinical trials right now, about how well they'll work, but there could have some effectiveness. They might not be as great as, for instance, a measles one. If there was something that says it's only 50% effective or 10% effective, would we want to actually use this? Is there any reason to or to not use it? I think I'm going to open this up to kind of both of you because I'm kind of curious from your different perspective. Would a stopgap vaccine be appropriate while we're waiting for one that's truly like the most effective one that comes online?
I mean, I think my thought is something is better than nothing. Obviously, first and foremost, we want a vaccine that's safe and effective. And, if scientists are able to successfully develop a safe and effective vaccine, even though right now it's only 50% effective, then that means that we're at least 50% ahead. And it gives all of us a fighting chance to mitigate the disease and to protect ourselves and our most vulnerable, while allowing researchers to continue to work on developing, other vaccines and more effective vaccines. I'm not sure if Dr. Hotez has additional
what do you
thoughts.
think about, Peter, of using these as like a stopgap, or even more, is it bad to have two vaccines if a second one comes online? Or is this going to start this weird market? I know we have a lot of unproven treatments that gets marketed all the time that science doesn't say work, but people do. We're going to end up having this unproven vaccine market where the vaccines just start giving out doses to patients that don't necessarily work.
Well, I think we're probably going to have to face the fact that our first vaccines out of the starting gate may indeed be partially protective vaccines. I mean, we've seen this now, for instance, with the adenovirus-based vaccine. It's not completely protecting. It's reducing clinical symptoms in non-human primates. But it's still resulting in a lot of virus in the upper airway. So it's not really probably won't inhibit transmission. So there's ideally, let me frame this a little differently. And if you think about a vaccine for COVID-19, an ideal target product profile, as we call it, would be to do two things. One, prevent you from getting sick, as sick enough to be in the hospital and be in the ICU. You'd love it to actually also protect against infection, meaning that if you get vaccinated enough people, you'll get herd immunity, so there won't be transmission in the community. It's looking that we have to face the possibility that the first vaccines that are out there, including things like the Moderna or the RNA vaccine, DNA vaccines, or adenovirus vaccines, may only do the former, may only protect you against getting sick and dying, which is still pretty important, right? That's what flu vaccines do more often than not. They don't halt transmission in the community, but they do seem to have a pretty big impact on reducing morbidity and mortality. But then as more vaccines come along, they might be better because they're stimulating neutralizing antibodies better, and then the first vaccines will get replaced. And there's actually a pretty good tradition of that. I mean, that's what happened with the rotavirus vaccine. There was a safety issue with the first one, RotaShield, and new ones came to replace it. Or the HPV vaccine, the cervical cancer vaccine, prevent cervical and other cancers, first had only got a few of the HPV serotypes, and then there was an expanded one that eventually replaced it. Or the Haemophilus influenza type B vaccine didn't work well in young infants, but it worked well enough in older children that it was still worthwhile. Then it got replaced by the one that worked really well in infants. So there is this. So I think there's a few lessons in all of this. Is one, we have to stop thinking about vaccines as a magic solution, which unfortunately it's couched in that. We're just going to all stay indoors, and all of a sudden the vaccine's going to come out, and then we can all go dancing in the streets. That's not going to happen. The first vaccines may not be the best vaccines. They may be partially protective so that they're not going to replace the existing health system that we have, but they'll use as a companion technology. Over time, in the coming years, and we think this virus could be around with us for a long time, possibly till 2025, according to some models, then we'll get new and improved vaccines as well. So it means that people are going to have situational awareness, recognize that our first vaccines are not necessarily our best. We might have some vaccines that work better in older populations versus younger populations. It's going to be a complicated and carefully orchestrated dance rather than how the Salk vaccine was rolled out, where they brought all the journalists to the auditorium at the University of Michigan and pulled back the black curtain like the Wizard of Oz, and then everybody clapped and got on with their lives again. It's probably not going to be that way, unfortunately. And then I think we also have to look at, once we do have vaccines that interrupt infection as well as reduce symptoms, can the vaccine alone achieve herd immunity? And now we're working with a group at City University of New York. It's a vaccine modeling group that was at Johns Hopkins and moved to City University of New York to see what percentage of the US population will need to be vaccinated in order to achieve that type of herd immunity if it's something that interrupts infection as well as disease. And we don't have the numbers yet. I mean, we're looking at it now. Hopefully it'll get published soon, or we'll put it up in bioRxiv. But it's going to require, I think, a pretty high percentage of the population get vaccinated. And as we can talk about, and I've been speaking with Rekha about this, is, will Americans accept the COVID-19 vaccine readily? When COVID-19, when it became clear COVID-19 was going to hit the US and hit the US hard, there wasn't many silver linings. Well, one of the few silver linings I thought was that, well, maybe this will finally put the anti-vaccine movement into hiding or in retreat, at least keep them at bay for a while. And that was true for about 2 weeks. And then they figured out how to get re-energized by this. And so now we've got some reports coming out from Reuters, did a survey, and the Associated Press has done a survey to show that a significant number of Americans, in part because of all the fake press releases by biotech and pharma and all of the mismessaging around warp speed, it's been playing right into the hands of the anti-vaccine movement. And to the point where now a significant percentage of Americans will not accept the COVID-19 vaccines, even if they come online, which is really concerning. So this is a self-inflicted wound that should never have happened. And it happened in part because of the way we message it. The anti-vaccine lobby likes to go up against both Rekha and myself and me, and they allege vaccines cause autism, and we've spent a lot of time refuting that. I had to write a whole book explaining why vaccines don't cause autism about my daughter. But then they also say vaccines are not adequately tested for safety. Vaccines are rushed. And there's this cozy relationship between pharma and the US government. Well, guess what's been happening in terms of the messaging and everything else we've been hearing about the last few months. So this has played right into the hands of the anti-vaccine movement and has been very enabling. So now we've got to do damage control and figure out a way to go, re-message what we're doing about the vaccine and go back to convincing the American public that vaccines are safe and not being rushed and all the other things the anti-vaxxers allege. And in the meantime, they've stepped up their attacks on me. I've got a new name now. I'm now the OG villain. It stands for the original gangster villain. As opposed to Bill Gates and Tony Fauci. I forget what they're calling them, but they were making COVID-19 virus in some secret lab in Area 51 or some crazy stuff like that. But, it's out there and it's gaining strength.
But Kirsten, I just want to just kind of interject and say that, I think that's why this event this afternoon is so important, to give, us an opportunity and scientists like Dr. Hotez an opportunity to kind of level set and walk through what the process is and what it looks like as to how vaccines are made, just to give the real world perspective and the true perspective that it is complicated. It does take time, and the experts and the researchers and the scientists want and need that time, because ultimately at the end of the day they want to put out a safe and highly efficacious vaccine for the public.
That's really great. I think it's a good conversation that we need to have. And I also want to know a little bit about, I think we're talking about rushing too much. And one of the concepts of actually speaking to the clinical trial process is this idea of a controlled human infection trial. So while you test a vaccine to see if it hurts the individual that gets it, you also need to make sure it actually works, right? So that means that they have to be compared to everybody else walking around to see if they actually get the virus get lower symptoms, and then other people who get it who don't have the vaccine. And we've had some commentators, even ethicists, as well as policymakers are advocating for this controlled human infectious trial, which means actively giving the participants in the clinical trial the virus. So exposing them to it and then really seeing the difference between that and people in the general public. I'm going to start with Peter. Can you kind of walk us through what this would be like, having a controlled infection trial? Why would we do this? Why would this be a good idea in your opinion?
Well, the idea behind controlled infection trials is that you vaccinate human volunteers, then you actually infect them with the virus. And there is a history of that for accelerating influenza vaccines, malaria vaccines, and we've done it with some of our parasitic worm vaccines. The issue around COVID-19, of course, is because this is a much more lethal virus. How can we do this safely, especially if we don't have a antiviral drug available to treat in case people get sick? And now, especially with all the thrombotic sequelae that we're seeing, the pulmonary emboli, the stroke, the coronary artery thrombosis, we don't really know what triggers that, what population is at risk. How can we do this with some level of assurance? And there's some controversy in the scientific community about whether we should proceed with this or not. I think the current thinking now is the fact that, creating a human challenge model is not quick business, because you've got to, first of all, produce the virus under good manufacturing practices, and that has to be signed off on by the FDA. That takes time. You also have to work out the dose of the virus and standardize a dose, so you're going to get the same result every time you infect a human volunteer with the virus in terms of number of virus particles that are there and the symptoms. You have to have good endpoints worked out, and you have to figure out the route of delivery. How are you going to do? Are you going to do this by aerosolized delivery or droplet? What's going to best produce the effects of human disease? The point is that takes time. It took us a couple of years to work out our challenge model for parasitic infection. I would imagine it could take the same amount of time for human challenge model. So there was a couple of interesting papers put out by people like Mark Lipsitch at Harvard recommending this, and Stan Plotkin, who's one of the giants in our field, has been enthusiastic about this. I think, though, the thinking is when you actually look at the nuts and bolts of what it takes to do this, it's not quick, and you may wind up getting an answer faster in a phase 3 challenge study than you would by developing the human challenge model. That's not to say we shouldn't try, and there's some thinking, well, maybe we should lay the groundwork for it in case we're not getting a quick answer by phase 3, but do it perhaps with a non-pathogenic coronavirus, because there are about 4 or 5 coronaviruses that cause upper respiratory infections that you could basically get started developing the challenge model using those viruses. In fact, it's been done before. So dust off those old protocols and get used to doing the challenge model. And then if there's a need to revert or to pivot to SARS-2, then you've at least laid the groundwork for it. But hold off on that until we know we need to do that.
So what I'm hearing from you is that we're not just going to take people and stick them in a room with somebody who's sick and have them sneeze all over them. There's actually a lot more involved.
Yeah, to get a standardized model that you can rely on for purposes of vaccine testing or drug trials, it's hard work, and it's doable. It's been done. I think our vaccine research center at the Baylor College of Medicine has actually done some of those studies. So yes, it's doable, but it's not quick work. It's not something that you'll have in a few weeks, and then we can get our answer on the vaccine in a few weeks.
And Rekha, do you have any other concerns about doing something like this, a controlled infection trial?
Yeah, I mean, I think Peter brought it up where we don't have a treatment for COVID-19, so we don't have an antiviral, and if you've got an individual who's in this clinical trial, exposed, intentionally exposed to the virus, we don't have the solution right now. So, that's something to keep in mind. And of course, as Peter clearly, delineated, that it's not a fast process. You've got to have very clear protocols and processes in place to not only, conduct those types of challenge trials, but also for the individuals that are in the trials themselves, and ensuring that there is very clear and robust informed consent, and volunteers who are part of these challenge studies clearly understand what is involved, what is expected, what is unknown, so that if they do choose to move forward in participating in this type of trial, that they clearly understand the risks associated with that kind of study.
So I'm going to pivot a little bit to talk a little bit more about public health. One of the things we've noticed recently in the whole space of vaccines is that we've seen a reduction overall in vaccinations. And I'm going to start with Rekha. How is the pandemic actually affecting overall vaccination rates, do you think, in the United States, Texas, but also globally?
Yes. So the short answer is the pandemic has had an impact on routine vaccination rates. There have been a lot of reports stating that, parents are hesitant to taking their children into the doctor's offices because of COVID. So it's not so much that they're concerned about vaccination. They're just scared about the pandemic and taking their child into a clinical setting. But as a result, we've seen significant declines in childhood vaccination rates, which then opens up another Pandora's box, which is if we start to see declines in measles vaccination rates and pertussis vaccination rates, then are we setting ourselves up for, a different kind of vaccine preventable outbreak? I've been talking to, immunization providers and stakeholders across Texas the past couple of weeks. And, they have consistently said that, they're hearing from parents in particular that they just don't know what to do, and they're looking for alternative ways to get their children vaccinated, but again, scared about the consequences associated with COVID. The other thing we're really worried about is, we've had children who haven't gone into the doctor's offices, especially smaller kids, so they're going to need to be caught up fairly quickly. We're out of school, but back to school will start in a few months, and so you've got young children who need to be caught up, children who need to have vaccines in order to get into school, and that comes on the heels of the upcoming flu season, which then also comes on the heels of a potential second wave of COVID. So I would not be surprised if we see a different kind of surge in our healthcare system in the latter part of the summer, early fall, because we're having to catch a large swath of children up to date with their needed vaccines.
Do you want to add anything, Peter?
Yeah, I mean, I am worried. I mean, we have seen this sharp decline in MMR vaccine coverage now. The CDC reported it. Starting to come back up a little bit now among the infants, but I worry it'll stay suppressed for a while, and then you fold in the anti-vaccine stuff that they're doing. I'm worried about a measles epidemic this fall. I think that's a real possibility, and then we're going to be in flu season. So, I've wrote a piece for the Fox News website about the potential of a triple epidemic, measles, flu, and COVID-19. And that's going to be really daunting if we have to deal with it. Hopefully that won't happen. But measles came back last year. Now rates are way down for a combination reasons now of the anti-vaccine movement and because of the emergency shutdown. Parents stopped bringing their kids in to get vaccinated. So yeah, I have real concerns about this.
I think also the pandemic is forcing all of us to think very differently in terms of how healthcare needs to be delivered, especially vaccination needs to be delivered, moving forward. We're hearing healthcare providers being creative and either having, certain days to have vaccination clinics. We're hearing, some clinics do drive-by vaccinations where, parents come up, one parent will, go into the office, check in the child, another parent's, in the car with the child, and then the nurse or the physician will come out, assess the child, vaccinate, and then, and then move on. So I think, we're gonna have to really kind of reconsider what vaccine access looks like, and the traditional way of delivering vaccines, may not be the right fit moving forward.
Okay, so I have one final question for both of you since we're almost out of time. And this is actually related to kind of like what you talked about, innovative vaccine, but also hype and misinformation that we see about science in general, but specifically during this pandemic and COVID. And I want to know from both of you what you think are good, reliable sources of information that our audience can go to keep up with the progression of just the vaccine, as well as in general information on COVID. Could you give us some suggested places that you go to, or perhaps a few that actually are easy for our lay audience to follow? So I'm going to start with Peter. So what do you recommend?
Well, for specific information on COVID-19, I enjoy looking at the models themselves, and there's three models that are out there that I know of that are pretty transparent, and you can look for it yourself because you hear about it on the news, but people don't realize you can look at it yourself. And this is one of the nice things about it is it's transparent. They explain their assumptions, sometimes not as detailed as you'd like, and some of them are fairly granular. So the Children's Hospital of Philadelphia policy lab models, they've got it by major county and metropolitan area in the US. So you can follow what's happening there. Then there's healthdata.org, the Institute for Health Metrics, University of Washington. And then University of Texas, Austin has put up models as well. And you can see that they have a lot of similarities, but differences in interpretation as well. And I think that's really helpful. I think the only one for Harris County is the Children's Hospital of Philadelphia one. And I just put that up on my Twitter site. That's a scary one. That looks like we're gonna start seeing a big return. But again, the models are based on assumptions, and the assumptions are not robust because we're still in a steep learning curve about the virus. In terms of vaccines, Nature magazine, Nature Drug Discovery did a nice overview a few weeks ago, although it's already starting to get out of date. BioCentury website has, they try to update their list of candidates, but it's amazing what you can go to just by going to BioArchive and just type in COVID-19 vaccines, and there's a lot of good stuff that comes up, and people like us are putting it up we're submitting our papers for publication, but to get the information out to the public, the journals are really good about having you put your information up on a preprint server. Now it's not peer reviewed, so there could be flaws, but at least it gives the opportunity for the scientific community to look at everything that's going on. That's how we got early information about the Oxford vaccine, the Oxford AstraZeneca vaccine, our vaccine, some of the others.
And Rekha, what about you?
So for COVID-related information, the Johns Hopkins University, their Coronavirus Resource Center is a great, one-stop shop to see what's happening, within the U.S. and globally. For general, healthcare information, Stat News is great. It's it's easily accessible and digestible. And of course, the Baker Institute has a fantastic blog that is updated frequently, and there's a lot of great information in there as well. And then as far as just general vaccine information, Children's Hospital of Philadelphia has a great website and vaccine resource center as well, and a lot of information about vaccines in general, about herd immunity, but also to kind of give guidance on how to talk to friends and family who may have questions about vaccines.
Also, I use for vaccine hesitancy, I like the Children's Hospital of Philadelphia site also. I also look at Vax Your Family. They've got some good information, vaccines.gov from the Health and Human Services. What else do I look? Vaxopedia has some good stuff, and that's by a doctor based in Texas who puts out a lot of good information. And there's always the Immunization Partnership website, which is pretty good.
Was the Immunization Partnership too?
Yes. Well, thank both of you for coming today. It looks like we're out of time, but we really appreciate you guys taking time out of your busy schedules to talk to us all. And perhaps we'll have time in the future to do another update if we need. In addition, I wanted to point out, as Rekha mentioned, you could go to the Baker Institute to find additional events on COVID. For the Center for Health and Biosciences, we do post weekly updates about what's going on with the COVID-19 data, which is authored by Vivian Ho. You should be able to find that on the Baker main website, or if you go to the center page, you can see the actual collection of it in its total, so you can watch the different weeks and see how things actually change as well. But thank you everyone for joining us.
Thank you.
Thank you.